Hello again. We're back to look at Chapter 5, and this time we're going to focus on proteins. I had to take a little break in my video making to eat a bunch of lipids. I had a big chocolatey bar. It was delicious. Okay, so we're focusing on these biological molecules that we're going to see inside cells. We've talked about carbohydrates and lipids, and now we're moving into proteins. Proteins are polymers. So think poppet beads again. Proteins are polymers of amino acids. So they're molecules, big molecules made up of many amino acids linked together. So let's write down that proteins, let's see if I can get this going, are polymers, polymers of amino acids. All right? Well, what are amino acids? Well, you could say that the amino acids are monomers of proteins, but that's kind of a weak definition. But let's just write it down. They're the monomers. They're the building blocks of proteins, right? They're the single units. And there are 20 different amino acids that our cells use to build all of the diverse proteins that we have. What does an amino acid look like? Well, every single amino acid has a central carbon. Bonded to that central carbon is an amino group. That's the, we have to learn our functional groups, okay? An amino group. Bonded to that central carbon is a carboxyl group. C double bonded to O bonded to OH, right? And a hydrogen. Bonded to that central carbon is also some other side chain. And that side chain can vary, so we put the letter R here in as a side chain. Now, sometimes we draw an amino acid in its ionized form. An amino acid in its ionized form, what happens is this amino group picks up an extra proton and becomes positively charged. The hydroxyl group on the carboxyl over here loses the hydrogen or loses a proton and becomes negatively charged. So we kind of have different ways of drawing an amino acid, but you should be comfortable drawing either of these. Now, each protein, remember, is made up of many different, 20 different amino acids, okay, connected to each other. And here's a picture of all 20 of these different amino acids that are used to build our proteins. When you go to biochemistry or an upper level class, you might have to memorize these. In Bio 120, we don't memorize these, but we can look at them, right? So you can see in each of these, the so-called backbone of the molecule of the amino acid is shown in purple. That's the amino, carbon, and carboxyl group. The side chain is shown in yellow, green, pink, or blue. And that side chain can vary. Some side chains are really simple, like glycine or alanine have very simple side chains. But here's the kicker. Side chains can be nonpolar or hydrophobic. So these side chains here are not hydrophobic. Look at all of them. There's a lot of methyl groups. There's a lot of carbon-hydrogen bonds, right? Sometimes the side chain can be polar, and that makes them hydrophilic. So you'll see side chains with hydroxyl groups or sulfhydryl groups or amino groups. Sometimes the side chains can even become electrically charged, and then they're definitely hydrophilic. So these different amino acids vary in their side chains. And each amino acid has a name. We give it a 3-letter designation and even a 1-letter designation. I know their names and their 3-letter designation, but I forget their 1-letter designations because I don't work with them that much. All right. Now, how do we build proteins? Well, we need to connect amino acids to each other. And if we want to connect amino acids to each other, what kind of bond has to form? Well, let's take a look. So what we've got in this diagram here is a picture of three amino acids, and we're trying to connect them, as you can see. And we connect them with a covalent bond called a peptide bond that forms between each amino acid. And as it forms, water is being removed. What kind of reaction is that? You can finish it. A dehydration reaction, right? What type of bond is this peptide bond that forms? Well, it's a covalent bond, just like these other ones that we've been looking at. It's strong. Okay? Where does the bond form? Well, it always forms in the same spot. I'm not going to try to write this out, but check out the pictures for a second. In this picture, we've got two amino acids, and we're trying to connect a third, right? In the picture, each amino acid is oriented in the same way, so that the backbone of the amino acid goes nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. The amino acids connect to each other at the carboxyl group of the first amino acid and the nitrogen of the amino group at the second amino acid. That's always where they connect together, and you can see their side chains are sticking out. It turns out then that because of this very rigid way of connecting and forming peptide bonds, the molecule has polarity. The molecule, oopsie, I wrote it wrong, has polarity. Sometimes we call the molecule a polypeptide instead of calling it a protein. It's made up of many amino acids linked together. It has polarity, meaning it has sidedness. I can tell which is the first amino acid. The first amino acid has the amino group sticking out. The last amino acid to be added has the carboxyl group sticking out. So the N-terminus represents the amino acid at the front end, and the C-terminus represents, this is the amino acid at the back end of my chain of amino acids. It's like poppet beads, really. Poppet beads have a hole at one end and a blob at the other end, right? So they have sidedness or polarity to them too. I can tell. I have to put them in the same way. I have to arrange them in the same way each time to add a new poppet bead. To add a new amino acid, you have to put them on in the same direction. So there's a first side and a last amino acid that are added. And what we're doing here by connecting amino acids is we're building a protein. It will become a protein. Where are proteins made? Does anybody remember where inside cells proteins are made? Da-da-da! What structure is the site of protein synthesis? Ribosomes. Right, ribosomes. We'll talk about them in Chapter 6. We're getting there. Okay. Now, proteins are not just a string of amino acids. They're not just boring like this. Proteins take on unique three-dimensional shapes. In one of the older textbooks, proteins were compared to knitting. And so I pulled out a really cute, tiny sweater that my mother-in-law knit for one of my kids when he was born. It's amazing. Right? It's complicated. There are different kinds of stitches, some with little holes, a ribbing stitch, a different stitch up at the top that I don't know its name to. In any case, proteins are a bit like knit sweaters. You start with a very simple yarn, a simple chain of amino acids, and you fold that chain in certain ways, and it makes a complex three-dimensional shape, just like knitting makes a complex three-dimensional sweater with different shapes. All right, so much for the knitting analogy. Apparently, this analogy was used for only a brief time in Campbell biology because back in the early 2000s, college students loved to knit. You would see college students just knitting. That was before cell phones. College students would just be knitting while waiting for classes to begin, and so it became kind of a popular analogy for a little bit. Knitting is actually good thing to learn, and it's fun to do. But let's talk about proteins. We can draw the protein. We can try to draw what a protein looks like in different ways. You know, we can use a ribbon model, or maybe a space filling model, or a wireframe. I don't use this one very much, but the idea is that proteins have these complex three-dimensional shapes, and structure influences function. Now, people who study proteins are like, we need to talk about protein structure. First, we want to divide it up. Let's talk about proteins as having 4 different levels of structure, and I love their names. Simple. The primary structure, the secondary, the tertiary, and the quaternary structure. Super easy. The primary structure is simply the sequence of amino acids. I'm just going to write AA for amino acids. The sequence of amino acids is the primary structure, and we've got a picture here of the primary structure of some protein, and it's got some numbers, and it's got little purple blobs. The purple blobs are the amino acids. The purple circles represent, glycine and proline and threonine and one other glycine. These are the chain of amino acids. How do I know that this amino acid is number 1? I mean, it's written there as number 1. This one way over here is the last one. I know that because amino acid chains have polarity. The first one laid down has the amino group sticking out. The last one has the carboxyl group sticking out. So here's the amino end. That's our first amino acid that's been laid down. There's the carboxyl end. Okay. The primary sequence is this chain of amino acids. But then you get more complexity. The secondary structure involves repeated coiling or folding. Coiling or folding of the amino acid chain due to regular hydrogen bonding that occurs within the backbone between hydrogen and oxygen, or hydrogen and nitrogen. So in the secondary structure, we see things like helical shapes taking place, or kind of a pleated sheet, like a paper fan you might make. What's going on here is hydrogen bonds form between the backbone of these amino acids. Not the side chains, but the backbone structure. And hydrogen bonds hold the chain of amino acids into maybe a helical shape or a pleated fan-like shape. And then we get the tertiary structure. This is even more complex three-dimensional shape. The language our textbook says is the tertiary structure are irregular contortions of the molecule that stabilize the 3D structures. This involves bonding between side chains. So you're going to have to write down bonding between side chains. I don't have room for it. We'll look at the quaternary structure next. This is a picture that shows us something about the tertiary. I just write like tertiary structure. Okay, so we've got this purple ribbon. That represents the backbone of amino acids in this molecule. And then we've got these different colored side chains. Oh, and side chains are forming different kinds of bonds. Hey, here's a hydrogen bond between 2 side chains. Here's, van der Waals interactions between nonpolar side chains. Here's a covalent bond that forms between these 2 sulfurs. Hey, here's an ionic bond. All of these bonds help to stabilize a complex 3-dimensional shape of the protein. And then there's the quaternary structure. The quaternary structure is when you've got 2 or more polypeptide chains interacting to form a protein. So we're going to write down 2 plus polypeptide chains. sometimes proteins, in fact many times, proteins are complex and involve more than just one chain of amino acids, but 2 or 3 or 4 all put together to form the protein. We say then that this protein has quaternary structure. Great example of quaternary structure would be in the protein hemoglobin. Hemoglobin is something that you're probably pretty familiar with. Well, sort of. Hemoglobin is a protein that's packed into our red blood cells, and it helps to carry oxygen and carbon dioxide actually too. It helps us exchange those, right? Okay. Hemoglobin is made up of 4 polypeptide chains. 2 of them are called alpha subunits and 2 are called beta subunits, and they have to be put together to make one hemoglobin molecule. Hemoglobin works really well, but if there is a mistake in one of these chains, we can have a disease called sickle cell disease. Because what sickle cell disease does is it leads to a hemoglobin that doesn't have quite the right 3-dimensional shape. It doesn't function perfectly. It works most of the time, hemoglobin, but then it doesn't work all the time. And what I want you to do is read the details about sickle cell disease and how it arises. It's pretty interesting, actually. Okay. So what do proteins do? Proteins are really important. There are lots and lots and lots of functions of proteins. I don't even have them written down here. I don't have a figure of them. Let's think about what proteins can do. Okay, sometimes proteins are enzymes. Enzymes help catalyze or speed up reactions inside our cells. We'll talk about them in Chapter 8. Some proteins are structural proteins that offer support. Maybe they're structural proteins that offer support in things like bones and in teeth and in hair and things like that. Maybe they're involved in transport. Some proteins help transport molecules, like hemoglobin does, or help transport molecules across a membrane. What else do proteins do? Oh, some proteins are hormones, like insulin. That hormone that helps us take in and use glucose is a protein hormone. Some proteins help in movement. You've maybe heard of actin and myosin proteins. They're involved in muscle contraction. Some proteins are receptors that sit at the surface of your cells and receive information driving cell behavior. Some proteins play a role in defense. So things like antibodies are proteins. Proteins do a ton for us. There are lots and lots and lots of functions of proteins. Okay. And they're very complex because they all have these unique 3-dimensional shapes. Now what happens if the 3-dimensional shape of a protein falls apart? Here's a normal protein, and it's this ribbon form. And you can see it's got this complex 3-dimensional shape. The ribbon itself represents the string of amino acids. We sometimes say that a protein can be denatured into a bad shape, something that's lost its 3-dimensional shape. Proteins can change shape when they're in strange conditions, like when they're when the temperature changes, or the pH changes, or the salinity changes in the environment, and that may cause them to totally lose their shape. And they might just be a ribbon, if you will. Do you think a protein that has lost its shape can function? Shape determines function. Occasionally, proteins that are denatured can be renatured. They can be folded back again into their 3-dimensional shape, that complicated 3-dimensional shape. We call that renaturing. Structure determines function, and we'll see this again and again, where protein structure, if it changes, will affect its function. So a protein enzyme that catalyzes a reaction, if it changes shape, it won't be able to catalyze the reaction. All right, we'll take a look at nucleic acids in the next video.